going to wait for, well, it's not 9 yet, but hopefully more people show up.
Yeah, I'm here early just because I have an exam to study for. I have an exam on Wednesday, and and then I have another whole course that I have to more or less finish, and I'm calling it mystery math. It's actually called Advanced Signal Processing. And when you know advanced algebra, linear algebra, with statistics a bit and with integration of some sort, and then mix it together and try to figure out a signal, it becomes mystery math.
Yeah. Yeah. Like, what kinds of topics are they covering in there?
We just covered frames, and that's expanded data sets so that you have dependent measurements in your matrices, but then you can kind of boil it down into a proper matrix because they're redundant, and you can, if you miss a signal, you can make up for it. Well, that's the idea of frames. And I have a project I have to look at atomic signal frequency to, what did he call it? Let's see. I'll get the book. This is the book. Foundations of Time Frequency Analysis. So I'm supposed to look at atomic time frequency analysis. And basically it's like, normally when you take a Fourier transform, your sine wave is kind of infinite time because it repeats itself.
Right.
And just like in Heisenberg and certainty principle, you have, if you've got short time, then you have, then your frequency is uncertain or something to that effect. It actually gets into some of that, but I'm supposed to look at a balance between the two so that you can sort your signals out. And it's called atomic for some reason. Wavelets fall under the category that I'm supposed to be delving into, but I have to start out big, and narrow it down until I get into wavelets. Wavelets, yeah, it's just wavelets, but he wants me to start with the basic huge theory first.
Oh, right.
Of course he does.
Yeah.
It's like, and I have to present on that in a week, and I haven't started. Why haven't I started? Because I've been too busy trying to do catch up with my other courses. I had to do a 10-page paper for the other course, so.
Yeah.
Anyway.
Yeah. It's pretty rough.
I don't know what. I have basically all the time in the world, and if they wanted to stretch this out, like I could do dedicate 3 days of the week to it and just work on it and then do what I have to do around here. But no, they want to stuff it all so that you're there's someone like me that's just like, why do I have to finish by the end of May? They said, oh, well, we have to prepare you like the first course is about electronics, basically.
Right.
And he says, well, when you go out into industry, they'll want you to know how to do this and that and the other thing. And I'm going, I'm not going out into industry. Like, what am I going to use it? Yeah, I'm going to use it on my thesis, but that's it.
Right. kind of hard sometimes, like if you're taking the course as, someone who's not like in a WE major or something to actually, because they usually design it for that major or for the, typical use case of the degree. So like, people who want to come from a different department or something to take the course. When I was in graduate school, for example, sometimes it's very hard to get like customized materials. Something that would be useful to you directly.
Yeah. Well, this is, what do they call it? They called it sensors and instrumentation. But I've taken that course at Red River College, and I've used it numerous times, so at least I understand the course. Ministry math is like, what? It's like, yeah, I went to write the exam, and I said, oh, I've faked math courses before, let's do it another time. And I passed it. I got I think it was 62%, and I sent the professor a note saying, well, I'm both very surprised and disappointed at the same time. Yeah.
Oh, well.
This is how to give Sharif a bad time, by the way.
Oh, well, I can't pass exams anymore.
Well, I just sort of going, whatever. That's where I'm at. It's like, whatever.
Yeah. Okay, well, while we got you, Susan, what do you think of the status of the axolotls?
What do I think of? Sorry?
The status of our project in Indian axolotls.
Well, the status of the project. Well, I need to get these stupid exams done. And I have to go change the axolotls after my exam on Wednesday.
You can change it?
Yes. They're at my brother's. He's feeding them.
Okay.
He says they'll need changing by the 24th or something, so I'm going to go change them. And I've got a number of beautiful females. And
Good. Do you have a nitrate kit?
I do, but what I've been doing with the axolotls is I've got two buckets that look similar, and I just change them from one to the other, so they get a complete change every 6 weeks.
Oh, okay. That's
Yeah.
working.
And it's working. Well, in the summer I'm going to have to put some Holt freighters in with them, because the summer water in Morden is deadly to them. I have an ultraviolet filter on the water there, but last summer I had a die-off. And it wasn't just me, it was the museum's salamanders too that were affected.
Oh. Okay.
Yeah, just way too much algae in the water, they couldn't take it.
Oh.
Yeah.
Some of those algae are toxic.
Yeah. Oh yeah. They're toxic, all right.
Yeah. Okay. Okay, Bradley, what's on today?
Well, we had Ujjwal was going to give a short presentation, but he can't make it. There's some internet trouble on his end. So I have a couple of things. Did you want to present something?
No.
Okay.
No.
I just had a couple of papers I was going to share with the group.
Okay, did you check your email? I sent you an email this morning.
Oh, okay, I didn't check yet.
Okay.
Well, as to the project with the axolotls, I'm just continuing it. Like it's,
Okay.
yeah, just sort of puttering along with it. That's.
Okay, good.
Okay. Oh, I saw your email now. Okay, so Stan is having problems getting reviewers for the paper.
Yeah.
Okay.
Yeah. Okay, that's it.
All right. Well, I can send him some more names, I guess. I can do a search.
Yeah, I sent that to Thomas Harvich also.
Okay. All right.
Okay. But I think from machine learning aspect, we kind of need to find people.
Yeah, so more machine learning oriented suggestions.
Yeah. Did you send Tom? Maybe Tom could do it.
Maybe. I can send him Tom's name and
Okay.
yeah.
Steve McGrew might be a good one too.
Okay.
Yeah, and I see our soap bar project scanner get funded.
It did not get funded?
No, I tried. I donated $100, but it didn't help.
Yeah, somebody gave us 100,000, and then it was no good.
Yes, they stole the credit card and thought it was a good cause and donated it. I suppose their heart was in the right place, but they shouldn't have stolen something in the first place.
Wow. Has that really happened? Is that like Kickstarter?
Yeah.
Oh, wow.
Yeah.
Yeah, we got $100,000, and then they took it away.
That's one of those, better spend it quick before it disappears.
Yeah.
Yeah, well, I'm only good for about $100, so I can spend it on electronics. Well, I recently bought these silly things. I don't know if you can see that or not.
Yeah, kind of. What is it?
It's a piezoelectric actuator from Thor Labs. And I thought they were a bit expensive because they were $90 instead of $60, partly because of the Canadian exchange. And then to top it off, they wouldn't put it in the mail. And so now I have a $40 bill from FedEx. And I'm going, great. But that's for my thesis project, so it's okay if I spend a bit of money on it, I guess.
Okay, Bradley, go ahead.
All right. Okay. Hi, Vinay. Okay. All right. Yeah. Welcome to the meeting, everyone.
Hello. Hi.
Hi. How are you? So yeah, like I said, Ujjwal was going to present something, but he has trouble with internet on his end, so we'll do that next week. I was going to update everyone on the Google Summer of Code. So we had we went through the OpenWorm Foundation for our applications. They're our sponsoring group, and then our organization is INCF, which stands for the International Neuroinformatics Coordinating Foundation. And so they are, they're the intermediaries between Google and OpenWorm, and they handle a lot of the administrative stuff. So thanks to them. And we had, we had 6 applicants for the pre-trained models project, and then we had 2 applicants for the Open DevoCell project. And we can't announce the winners until, I think, the beginning of May, so we're not going to do that yet. But we have to say that we had some very good applicants for both projects, and it was a very tough decision to make as to who to pick. But we did pick 2 people, one for each project. And so the people who don't make it, they're going to be invited to join the group if they want to, participate in the meetings, the Slack channel on the OpenWorm Slack, any of those things. They're welcome to do it. And one person from the Open DevoCell group, the person who's not been selected, will probably be invited to participate in the Google Season of Docs, which is a new thing Google has, and it's in the fall, so it's not during Google, Summer of Code. It's the fall season of Docs, so it's more documentation oriented. And we did this last year in OpenWorm, and it was pretty successful. I think we built a documentation system for OpenWorm. Well, I should say the the person who got the award did, and I co-mentored someone through the process. So now we have this great automated documentation system. So we could do, we can do the same thing in DevoWorm. I have to still work on the application part on our end, but once that's done, we can invite that person who wasn't selected for the Open Devo Cell project to work on that, and they would just have to modify their application accordingly. So hopefully, everyone is, everyone if they applied to the GSOC and didn't get selected, then they're invited to come into the group and participate. We've had a couple people already come in, applicants who are participating, so that'll be, that maybe we'll have some new people showing up. So Ujjwal is next week, and then that GSOC announcement is I think the week after. Also, let's see. I think that's all the news I have. Vinay, what's going on your end? Is there anything interesting?
Hello?
Yeah.
Yeah. Pretty much, it's the same. Like I'm in my final semester of my bachelor's, so we are working on our bachelor thesis. So that is happening right now, and also I'm doing an internship at Philips, based on medical, it's like an analytics project that I'm working on as part of internship.
Okay.
So both those things are I'm busy with this in this quarantine period. I'm just busy with both of those. That's it. Nothing much interesting apart from that.
No, it's good though. Susan's got a lot of coursework she's working on. We were just talking about that. And then in terms of the axolotl data, since we have Susan and Dick here, both, still kind of sitting on that for right now, thinking about how to analyze it. I haven't had too many discussions about it yet, but we'll be getting there. I sent out last week I sent out the paper on the Google embryo. I don't know if people saw that email, but the paper that Dick wrote on the vision for the Google embryo. So it's like Google Earth. It's just the surface of the embryo, and so you can. That's like the ideal way that we might visualize the surface of the axolotl embryo. And it's going to take a lot of work. I think it's going to take the right solution to do it, but I think it can be done. I'm optimistic. So Dick put something in the chat here. This is something that's just yes, okay. So he's working on a paper here. It's Richard Gordon and Natalie Gordon. So Natalie Gordon is his wife, and she doesn't come to the meetings, but she's in the house with him there. Let's see. When we were all triangles, our extraterrestrial-shaped droplet, polygonal archaea origin of life. Okay, so this is in preparation. I'm working on this now. Let me know if you're interested. So that sounds pretty interesting. When we were all triangles. I don't know if I've shown you the stuff I was working on with what do I call it, multicellular systems or not, but that might actually be relevant to this. I don't know, you'll have to give us maybe in the next couple weeks you'll have to give us a short or maybe I'll give the discussion on that, on the multi-cell systems, because they basically use a bunch of shapes, and the shapes replicate, and then you're able to see you can actually use mathematical tools to see sort of like how they're, evaluate whether they're different groups, whether they're different modules. So you have a lot of triangles that are aligned in a structure, and you use a line segment to draw through the edges. And if they complete an Euler circuit, then they're a unified system or module. And if they don't, then you break the thing into two modules and you retest it. And it's the idea that you want to be able to have like exchange between cell membranes to get things in and out. So the idea is you would have this Euler path that would tell you whether it was a unified structure, if it had like paths between only one path between each membrane, and they would, that's the idea behind it. And it's really abstract and kind of out there until I actually show you the experiments. But I can do that in the coming weeks. Maybe, I don't know, maybe it's relevant to this, but it looks interesting. I know that a couple of years ago you had that book on habitats, and we had a paper for that, but it wasn't really suitable for that, so we ended up publishing it in Biosystems. And that was a pretty good book. This looks like it's kind of a follow-up to that kind of work. I mean, so yeah, well, I don't know if I have it right now. I don't think I can get to it now, but anyways, so that's another thing. Again, if you're interested, let Richard know. You have his contact information. Okay, archaea are single cell prokaryotes, many polygonal. I'm sending Bradley a background paper. So he'll send me a paper, and then I can maybe put this in a little bit more context at the next meeting, or we can discuss a little bit about the idea. I don't want to share the paper prematurely, but I'll probably present, maybe we'll present on something related to this. So yeah, well, send me the paper, and maybe we'll have a private discussion about it. Let's see. So today I was going to cover a couple of papers that I haven't had a chance to cover in meetings past. We had things that we were talking about. I can't remember. Oh, last week we were talking about the John Conway memorial tribute. So John Conway died like 2 weeks ago now, and we had some interesting things on John Conway's work on his game of life. So I was showing them to the group. This week, focus more on new developmental papers. A couple of really interesting papers. I don't know if I understand them all, but I'll go through them for everyone. So let me share my screen here. All right. Can you see my screen here? The papers repository. Okay. This one. Okay, this one. Okay, yes. All right. So the first paper is this. This actually was something I found by one of these news press releases, and they made it sound really like, the press releases make it sound really super provocative. It's almost like science fiction. And then you read the paper, and it's still pretty good, but it has like, it's a little bit difficult to see how they got the news release story out of the paper. But, that doesn't take anything away from the paper. That's just an observation. Anyways, this paper is Emergence of Collective Oscillations in Adaptive Cells. And I know we've talked a lot about differentiation waves and things in cells in this group. And so this is a sort of an overview of this set of experiments this group did. on collective oscillations and cell, I believe it's cell membranes. So, collective oscillations of cells in a population appear under diverse biological contexts. Here we establish a set of common principles by categorizing the response of individual cells against a time-varying signal. A positive intracellular signal relay of sufficient gain from participating cells is required to sustain the oscillations, together with phase matching. The two conditions yield quantitative predictions for the onset cell density and frequency in terms of measured single cell and signal response functions. And so they use mathematical modeling to show that cells adapt to a constant stimulus, and it develops a leading phase in an active frequency window that enables cell to signal energy flow. Analysis of dynamical quorum sensing in several cellular systems with increasing biological complexity reaffirms the role of adaptation in powering these oscillations. And so they argue that the cell communication channel is generally dissipative. And we talked about that in George Mihailovsky's presentation. He talked about dissipative systems or dissipative structures. And then, oh, now it's having a problem loading more pages for some reason. Oh, this is the wrong one anyways. Okay, so I don't know why I can't open this paper. Let me try to open it with a PDF reader. Maybe that's the key here. Okay. All right. I misspoke a while back about the membrane. The membrane is the other paper, but this is just a general cell signaling mechanism. Okay, I think we're going to work here. I think it's going to work. All right, I think we're up. There we go. So this is Nature Communications. That was the abstract. Now if we get to the paper. There we go. So this kind of shows you this. Oh, we've got another person coming in. Okay, there's Ujjwal. Ujjwal. All right. So, hi. Actually, I'm presenting papers right now. Are you still planning on presenting today, or do you have your slides, or you don't have enough bandwidth on your end? Okay. Oh, okay, that's fine. So I just pulled up this paper on collective, like cell-to-cell communication with collective oscillations. So this is a population of cells, and they're exhibiting this cell-to-cell signaling mechanism. And so here's their model here. I don't know if I can zoom in on this. I guess I can. Okay. So this is their model. Spontaneous oscillations in a communicating cell population. Scenario mechanical oscillators where cells communicate via a shared displacement S of the physical environment. And so this is the shared displacement, this parameter S, and they're using a spring here to show the displacement. It goes back and forth here. Activity A of a cell against the displacement is regulated by an intracellular network which responds to S through a mechanical sensor. So this activity in A is regulated by this network, and then it responds to S, so S is stimulating the cell. A is this activity, which is driven by these biochemical networks. And then that's the thing that's regulating so it's regulating this whole interaction. And then B is down here is an illustration of chemical oscillations where cells interact via a shared extracellular signal S. The signal is sensed and secreted by individual cells. So this is the S, this is the signal concentration. So this could be any number of secreted proteins or morphogens, as they call them, or whatever. And they're secreted by the activity A of these biochemical networks. They're secreted into the extracellular matrix, which is the area between the cells, and then they communicate with the cells. So these S's are related, I suppose. The shared displacement is the displacement of the cell by this physical environment. And then the S here are these signal concentrations in the physical environment. It's not clear exactly what the exact relationship is, but they're both, I think they're related, is what they're saying. So you have this sort of forcing mechanism on the cell. It's doing something inside the cell. It's forcing it to adapt to its conditions. And then it's exporting some signal, and it's telling the other cells that there's something going on in the environment. Now, this is all going on in parallel, so you can see that when you have this sort of relationship, it's sort of a collective behavior type thing where, the cells act in parallel, they inform their neighbors. It's sort of like a cellular automaton where they're informing their neighbors about what's going on, and then they're adapting likewise. And the idea is that they can coordinate this behavior so that they behave in certain, coordinated ways. So they talk about homogeneous cell populations are able to exhibit a rich variety of organized behaviors. Among them, periodic oscillations. So periodic oscillations are oscillations that are coordinated but also periodic in time. So, if you see a bunch of cells go from red to blue and then back to red again, having that sort of a very orderly transition amongst all cells, that's a sort of coordinated, organized behavior. Then they talk about during mound formation of starved social amoeba, cyclic AMP waves, and those are things that are in the cell, guide migrating cells towards a high-density region. Elongation of vertebrate body axis proceeds with a segmentation clock. Multicellular pulsation has also been observed in nerve tissues during dorsal closure in late-stage Drosophila embryogenesis, and more. So they give a bunch of examples here. In these examples, communication through chemical or mechanical signals is essential to activating quiescent cells. Dubbed dynamical quorum sensing, to emphasize the role of increased cell density in triggering the oscillations, this class of behavior lies outside the well-known Kuramoto paradigm of oscillator synchronization. So this Kuramoto paradigm is it's a special type of model for modeling oscillatory behavior. It's a set of differential equations, and the place it's most famously been observed is in firefly colonies. So if you're familiar with Stephen Strogatz, as a mathematician, he wrote a book called Singh. This is about 20 years old. And in that book, he gives a good sort of a popular science example of fireflies synchronizing their flashing. So if you are familiar with fireflies, they're out in the summertime in the grass, and they fly above the grass, and they give off this glowing signal from their tail, and it's a luciferase protein, so it glows when they their nervous system sends a signal to the sac of luciferase, and they can do this. They can synchronize their glowing so that they're glowing in some sort of orderly pattern. And so they observe their neighbors and they do this sort of glowing. There are other ways in which they regulate their fire, their glow, their fluorescence, but that's one way. And so they can basically communicate with one another and synchronize this. And it's been modeled using this Kerr-Moto oscillator model. And so this is what they're proposing, that this sort of behavior lies outside of that paradigm. Dynamical quorum sensing, I'm not familiar with that, but I do know about quorum sensing, which is, you commonly see it in bacteria where they have to, meet a quorum in their social group to, do some sort of behavior. So they execute a behavior based on what they're sensing of the neighbors, what their neighbors are doing, and then they make a decision to do things in unison. So that's what they're kind of proposing for the cell populations, that they're coordinating their activity in this way and that they're acting in unison. So they go through some equations here. So they give some necessary conditions for auto-induced oscillations. There's some equations. I'm not going to walk through them because I really haven't dug into them to see, what the relationship is between them. But they think of it in terms of an equilibrium state of S. So the equilibrium S, we talked about S up here. It's this sort of communication channel between the extracellular space and the cell. So the S is either both forcing the cell and a signal concentration from the cell, and it's mediating all of this. And so when it's out of equilibrium, or when you can disrupt the equilibrium, you can sort of get this sort of quorum sensing to happen. Let's see what else is in the paper. And so they really go through this. Talk about cell to signal energy flow. Adaptive cells shows phase leading response. So they kind of walk through some of the basic models of chemical oscillations. They talk about adaptive cells showing phase leading responses. So this is a type of sort of regulation here. Phase leading response to a low frequency signal has also been reported in the activity of E. coli chemoreceptors and in the osmo response in yeast. And so these are examples of adaptive sensory systems whose response to a step signal at t-0 is shown in figure 2a. So this is the figure 2a. So this is all basically the dynamical response of the signal on the cell. So the signal is sensed by the cell, and the biochemical networks respond. And they respond in a certain way. It's sort of like sort of a cellular version of signal processing. So they're getting this signal and they're trying to determine how to behave. But because you're getting, sort of a lot of, like it's based on a concentration, you have this sort of response that the cell has to give. So there's some sort of profile that the cell has that you can measure that's based on the concentration of what's outside the cell, on what the cell actually does inside. What concentrations of this S are leading to some sort of triggering behavior in the cell. And so that's what they talk about here. So A is response to a stepwise signal. So if you provide a stepwise signal based on S over time versus this, this is another type of signal that they're using. So response to a stepwise signal. After a transient response, A returns to its pre-stimulus state with a small error epsilon. So this is the error. So this is the response curve. This is the stimulus curve. So A here over time there you see this increase in activity, and then it sort of returns to its baseline, but with a little bit of error or a little bit of what you might call hysteresis. In the simplest case, the transient response is controlled by the activity shift timescale and the circuit feedback timescale. Solid and dashed lines in here correspond to the overdamped and underdamped situations, respectively. So this is the solid line is an overdamped response. The dashed line is an underdamped response, and the damped response is it's just damped. It's it's not it's sort of inhibited in some way. Response to a sinusoidal signal at low and high frequencies. So this is B. This is the response to a sinusoidal signal as input. So S over time is this red signal. This is the sine wave. That's the thing that's going into the cell. And this blue function is the thing that's the cellular response to that forcing. So you can see it's dampened a bit in terms of its amplitude. And then this is phase lead, and then this is phase lag. So that now you have this S, which is the forcing mechanism, this red sine wave, and then you have blue, which is the response of the cell, and that is actually lagged a bit rather than dampened. I mean, it's a little bit dampened, but it's not. It's mostly lagging here. And so that's part of their model here. And they argue there are excitable dynamics. So these DQS, these dynamical quorum sensing systems in dictyostelium, which is a slime mold, and other eukaryotic cells take the form of pulsed release of signaling molecules. The highly nonlinear two-component Fitzhugh-Nagumo model is often employed for such excitable phenomena. So Fitzhugh-Nagumo is a oscillation mechanism that's sort of based on neuronal models. So Fitzhugh-Nagumo is it's kind of like the Kuramoto model. It takes into account a lot of different cells, except it assumes that they have action potentials, and those action potentials, like in neurons, are synchronized. So I mean, all cells have they have electrochemical signaling, but they're assuming some sort of action potential, which is limited to neuronal cells, and they're assuming that they assume that with an action potential that you have this stimulating, this forcing mechanism, and then there's a response by the cell, and this response is one of these types of functions that you see here. Action potential is very distinct. It has like a couple of components to it. So it has it rises and then falls and then comes back to an equilibrium. In this case, you don't really see that, but they're using that model to get at this mechanism. So they often, people use this Fitzhugh-Nagumo model for a number of excitable phenomena. So when they say excitable, they mean something like an action potential, something that would, like a cell would be excited by a stimulus. Similar to the sensory adaptation model discussed above, each FHN circuit has a memory node Y that keeps its activity at a low, or A, keeps its activity of A low, which is the parameter A, the resting state, under a slow varying signal S over time. So as the signal S over time stimulates the cell, there's activity in the cell, but it's kept low as a resting state. On the other hand, a sufficiently strong noise fluctuation or a sudden shift of S, which is that forcing mechanism, sends the circuit through a large excursion in phase space, known as a firing event. So if you have this sudden pulse to the cell, the large excursion in phase space is basically this sort of action potential-like thing, where you have this large response and then a return to more or less equilibrium. And so they talk about this in figure 4, and this is figure 3. And then figure 4 is here where they have a little bit, they talk about the single cell response some more, and they show some of these are computational experiments. So they're doing a lot of this, I think, in the absence of like recordings, but actually I think they may have some recordings in here, but they're showing this in terms of like simulation versus theory. So this is a pretty interesting paper. Like I said, I'm not going to go through much more of it because I don't know if I could really do it justice, but they talk about these glycolytic oscillations in yeast as an example of this type of quorum sensing. And then, yeah, so I think, and then they go back to dictyostelium a bit. It's a very good paper. I mean, it's a little over my head actually, but it's a nice example of how cells communicate and how they communicate to different signals and how they collectively respond to that. Okay, so we had a little disruption here. Okay, so that's that paper. Then this is the next paper here. Topological turbulence in cell membranes. So this is a nice one. This is a Nature Physics paper that just came out recently. And again, this had one of these press releases attached to it. So, the press release, actually the press release and the paper were both equally as inspiring. So I like this paper quite a bit. And this actually is more directly relevant to the stuff we've been doing with differentiation waves. So topological defects determine the structure and function of physical and biological matter over a wide range of scales, from turbulent vortices in planetary atmospheres, ocean of quantum fluids, to bioelectrical signaling in the heart, and brain and cell death. Actually, that's heart and brain and cell death. Many advances have been made in understanding and controlling the defect dynamics in active and passive non-equilibrium fluids. Yet it remains unknown whether the statistical laws that govern the dynamics of defects in classical or quantum fluids extend to active matter and information flows in living systems. So they talk about we show that a defect-mediated turbulence underlies the complex wave propagation patterns of Rho-GTP signaling protein on the membrane of starfish egg cells, a process relevant to cytoskeletal remodeling and cell proliferation. And so they talk about this in very physical terms. So they talk about several key statistics and scaling laws. They use a Helmholtz Onsager point vortex model, as well as a generic complex Ginzburg-Landau continuum theory model, suggesting a close correspondence between the biochemical signal propagation on the surface of a living cell and a widely studied class of two-dimensional turbulence and wave phenomena. So that's a mouthful. But the basic idea is that they're looking at these complex wave propagation patterns on the membrane of egg cells. And so they're actually looking at this from a physical standpoint. And so topological defects are persistent discontinuities in the order parameter fields of continuum physical systems. And so these defects critically determine the structure of fluids, rigidity of solids, and the dynamics of biological matter. A particularly interesting class of dynamical defects that has profound implications for morphogenesis and complex biological functions underlie the formation of spatial wave patterns and reaction diffusion systems. And so if you're familiar with the Beloutov-Zhabotinsky, the BZ model of morphogenesis, it's actually a model of chemical reactions, but sometimes you use it to understand morphogenesis as well. This is kind of what they're going at here. So there's some figures here. So this is a typical BZ reaction, this image in E. So this is a picture of what it would look like. So what they do is they pour some chemicals into a vat, and they take an image of it, like how the interactions between different chemicals happen in this. So it's a reaction, a chemical reaction, and there's all this pattern formation going on. And then actually what they're doing here is they're showing defects in the pattern formation. So this is alright, so this is topological defects populate the phase field of rho GTP waves. So A is membrane rho GTP waves are visualized with GFP and RGBD reporter and live starfish oocytes. So that's A. And of course you have the where they're showing the biochemistry here, and then they're showing what the pattern will look like on the surface of the egg. Then B, representative snapshot of rho GTP waves from 4 oocytes of different steady states. So this is B, where they have different patterns that they've observed using microscopy. C is normalized intensity cross-correlation plotted as a function of distance in the 4 states shown in B. Error bars represent standard error throughout time, so that's this graph here. Then D is representative time series of fluorescence intensity at the single pixel level for the 4 states shown in B. So that's this one here, which is the sort of shows the signal intensity over time for these 4 images. E is a representative phase field reconstructed from pixel oscillations, or as a dense population of topological defects. So this again is a phase field view of this surface, and like I said, it looks like a Belousov-Zhabotinsky reaction, but this is actually an image of these pattern formations shown in B. This is just a different view. They reconstruct the fluorescence of these images using a phase field reconstruction, and then they look for defects. And so they find some defects in these boxes here. And so then F is time-lapse snapshots of the phase field in the vicinity of a topological defect every 10 seconds. So this is showing it broken out in time in terms of this reaction. G, trajectories of defects undergoing creation and annihilation events. So this is where they have creation and annihilation events. I don't know how exactly they do these. They do this analysis, but they're showing the defects in this field, sort of in this figure, and they're showing how sort of it moves. So the defects, I guess, are short-lived. I can't really tell, but both types of events always involve pairs of oppositely charged defects, red being the positive and blue being the negative. The defect density fluctuates around a constant value at steady state I. Characteristic wave numbers are positively correlated with defect density for all 11 states. Error bars represent standard deviation. Dashed line is the line of the best fit. So that's for H and I. So this is what those graphs look like. Vortex dynamics in the phase field velocity field. So this again is another analysis of these, the analysis of the vortices. And this is a comparison of what they see experimentally with a discrete Helmholtz on-Sager point vortex model and a complex Ginzburg-Landau continuum model. So they use these 2 different models One is a point vortex model and the other is a continuum model. And they basically, just compare the two models with the empirical observations. And then that's pretty much the paper. So I think next week I'm going to present this in maybe a little bit more detail. This is the paper I really wanted to show people, but it didn't really give it a lot of time. Didn't really have a lot of time allotted. We have a technical difficulty here. So I'm going to present this a little bit more detail in future meetings. I will send these papers out because we didn't have a lot of people at the meeting. I'm going to send these papers out in an email, a link to the repository where I have the papers. And I think this is a nice paper, but I'm not sure if I totally understand what's going on in it. It might be relevant to some of the stuff we're doing with differentiation waves. We'll see. Maybe. And then, of course, the other thing we're going to talk about is the work on the multicellular systems that I told you about. That Richard is working on a paper on triangular cells. So we'll see how that all comes together. So thank you for attending, and see you next week. If you need anything, catch up with me online.